So in our last class, we talked about the entire process of fertilization in the oviduct, the um, mitosis of the zygote to form a small embryo, implantation in the wall of the uterus, where the placenta forms, and then the growth and development of the embryo and the fetus inside of the uterus. We talked about labor and delivery somewhat. Did we do the slide? I can't remember where we were. Any idea? Did we talk about labor and delivery completely? We did. Okay. So Jen, just to summarize sort of that process, labor is the contractions of the uterus, the involuntary contraction of the uterus, which start to move the fetus down lower into the uterus, where it presses on the cervix, okay, which eventually has to dilate and kind of open up to like 10 centimeters or so to allow the fetus to move through it. And then from there, labor moves into the delivery process, in which the mother is pushing the fetus out through the birth canal, through the vagina. And then after that, in the final stages, the placenta breaks away from the uterus and the placenta comes out as well. There are times, however, when there's difficulties in the childbirth. Sometimes, like for when my daughters were born, they kept their heart rate. So when a mother is in labor and she's there, they hook her up to a fetal monitor, which monitors the heart rate of the fetus, the oxygen level of the fetus. And so when my daughters were being born, they all had like a relatively short umbilical cord. And so what happens is sometimes it gets stretched, or it could get sort of kinked, or it can get wrapped in a certain way that is not delivering enough blood to the fetus. And so sometimes we could watch as my wife's in labor and the heart rate of my daughters would drop down, or the alerts would go off. And so sometimes they'd rush in and the nurse would kind of turn my wife over to try and move things around so there's less pressure on the umbilical cord. And it was never a major problem. But sometimes it is a major problem. Sometimes that umbilical cord could sort of be wrapped around the fetus's neck, or it could be kinked. There's lots of things that could happen. And so in those situations, they may have to do things to either speed up labor and delivery. So there's certain hormones they can inject into the woman that's in labor, and it'll speed things up. Or they may have to use special tools to help deliver the baby if it's not moving through the cervix and not moving through the vagina quickly enough and it is in distress. They might have to use special tools. They have suction tools and like salad tongs and things. They help to remove the fetus as it's being born. Or sometimes if there's major issues, they might want to get the fetus out as soon as possible. And that's when they might do a cesarean section. And so in a C-section, rather than the fetus being born vaginally, they do an operation and they make an incision in the mother's abdomen through her skin. She usually has like a spinal block, so she's still awake and aware, but she has no feeling from like, shoulders, or not shoulders, like chest down. So they can do the surgery. And so they cut through the skin of the abdomen, they cut through the abdominal muscles. Then below that is the uterus. They have to cut through the uterus. Then is the amniotic sac, which they might have to puncture. And then they take the fetus out through this incision, and they can do all this pretty quickly. So, sometimes labor and delivery might take, 10 hours, 12 hours, 24 hours even. And so if there are issues going on with the fetus, they can do a C-section in, 20, 30 minutes from prep to having the fetus out. So they can do that if the fetus or if the mother is under severe stress. They try to avoid it as much as possible because the recovery from a C-section takes a lot longer than a vaginal birth. So it's a little bit more, traumatic on the mother in terms of her recovery and so forth. But they'll do it if necessary. Okay. Any questions about labor, delivery, birth, C-section? I'll still come back to these videos maybe. Let's talk about a couple of things, other like random birth topics. Twins. There's different types of twins: fraternal twins, identical twins, conjoined twins. What does it mean? What are fraternal twins?
They don't look like, so it could be like a guy and a girl.
Okay, so I'm gonna correct that a little bit. They might look alike. Right. They don't have the same exact DNA, but they can look very similar. Like there are times when fraternal twins, we might mistake them for identical twins. Like if I look at pictures of my daughters when they were 8 years old, like sometimes I'm like, I don't know, is this Alyssa or is this Olivia? I can't tell the difference between them because they share a lot of similarities, right? You might look very similar to a sibling. And so fraternal twins, they don't have the exact same DNA, but they're still brothers or brother and sister or sisters. So they still might look really similar, but their actual DNA is not identical. How do they, and they can be different sexes. How do fraternal twins form? Why might someone be pregnant with fraternal twins? Do they even know the process that happens? Ash? Yeah. Sometimes, so in a typical reproductive cycle, a woman might release a single egg from one of her ovaries, and it can get fertilized by a sperm cell, and she can get pregnant. Sometimes, however, both ovaries might release an egg at the same time, and if they both get fertilized by sperm cells and they both implant, then there's going to be two babies there. But they came from two different sperm cells, two different egg cells, so they're just like brother and sister or sister and sister. They're not any more similar than siblings are because they came from different sperm and egg cells. That's how these, and that's why they can be different sexes as well. So, and sometimes a woman might release 3 eggs, and you could have triplets, or even more. Now, sometimes if a couple's having difficulty conceiving a child, the mother may take certain medications that's going to encourage her to ovulate more regularly. Well, sometimes those hormones can almost work too well, and she might release 3, 4, 5, 6 eggs at once. And if they all get fertilized, then we're in the realm of like septuplets or something like that. So yeah, that's fraternal twins. How are identical twins? How are they formed then? And what does it mean if twins are identical? Jason?
Isn't it the same egg?
Same egg and same sperm cell. So in some cases, an egg cell is fertilized and it starts to divide into a little embryo. But sometimes, for some reason, the embryo splits in two. And then each of those starts dividing. And they each start to form their own independent embryos. But those two embryos, they came from the exact same sperm cell and the exact same egg cell. Therefore, they have the exact same chromosomes. They have the same DNA. They're always going to have the same sex because of those chromosomes. So identical twins share all of their genes with their sibling. And so that's the way identical twins form. A single zygote, for some reason, splits and forms two identical zygotes and embryos and so forth. There are times when that happens, but the two zygotes that split actually don't fully separate and they're still sort of touching. And when that happens, conjoined twins can form. Twins that actually physically share a part of their body. And wherever those eggs were touching, they're always going to share like part to like part. You know, it's abdomen to abdomen or chest to chest or head to head or something like that because of the way the embryos form. And so conjoined twins sometimes can be separated from each other, but it really depends on how much of their vital organs they actually are shared between them. There's times when conjoined twins can be formed connected sort of at the chest, but only have a single heart. And obviously they can't be separated in that case because they only have one heart between the two of them. So it just sort of depends on how they form, whether or not they can be separated. And we see some examples of these twins here. In terms of birth control or contraception, avoidance of pregnancy, or sometimes people are trying to couples are trying to get pregnant and might monitor. One method is to monitor the reproductive cycle. As we talked about, through the female reproductive cycle, pregnancy only can happen within a window, right, around the time of ovulation. So sometimes people are tracking their reproductive cycle and trying to figure out when they're ovulating, because if they have sex when they're ovulating, it's going to increase their chances of getting pregnant. So there's even like a urine test that women can do that kind of tells them when they're ovulating, so that they can be more likely to get pregnant. At the same time, some people also try to monitor reproductive cycles to avoid pregnancy. Okay, and if they can avoid having sex during that time, then it's possible to avoid pregnancy. It's not a very reliable method of birth control because there's a lot of variables involved. Sometimes a person's reproductive cycle is irregular and they're not ovulating on a predictable scale. Sometimes the egg cell is viable for several days, and sperm cells are viable for several days. So the window, it's not like, if you have sex only on day 14 of your menstrual cycle, that's the only time you get pregnant. That window is a lot larger than that. So sometimes it's not a very effective or consistent means of birth control. Lots of other ways. Birth control hormones, like an oral pill that women can take, can stop her from ovulating regularly, so that if she's not ovulating, obviously, then she it's not going to become pregnant. That's one way. There's also IUDs. Is an intrauterine device. It's a device that a doctor will insert into the uterus, and it prevents a woman from getting pregnant. That's another common, and that's sort of like it's not permanent, it's not forever, but it's a longer lasting. So with like oral contraceptives, a birth control pill, a woman has to take pretty much every single day consistently, otherwise it doesn't work very well. Like an IUD is a device that's inserted and it lasts for 5 or 6 or 7 years, and then it can be replaced afterwards. It can be removed if a woman then decides she does want to get pregnant. So it's a common means of sort of longer term contraception. Obviously, barrier methods include things like condoms, which prevent sperm from actually reaching the egg cell. There's chemicals. Spermicides are chemicals which actually destroy sperm cells so that they're not going to be able to get to the egg. And then there's also hormonal treatments that are longer lasting. There are hormone injections that women can have that's going to give them sort of longer term stop to their ovulation. There's all sorts of different types of sort of hormonal birth control methods. There's also permanent means, which are vasectomies in men, which are pretty much permanent, where they cut the vas deferens, seal it off so that sperm cells aren't released from the testes, and therefore women can't get pregnant. And the equivalent in women is a tubal ligation, where the fallopian tubes are cut and sealed so eggs aren't released and can't get fertilized. But those are pretty much permanent means of contraception in which somebody says, I don't ever want to have children anymore. Any questions about any of those? And the other thing is lots of these prevent pregnancy, but not necessarily sexually transmitted diseases. So if a woman's on a pill, or if a man gets a vasectomy, it can still transmit and receive sexually transmitted diseases. Things like condoms can prevent that. And this is interesting, sort of effectiveness, right? Like the least effective form of contraception is like monitoring a woman's cycle and trying to time sex with when they're ovulating or not. That's only like 24% effective. So that's not great. So this is for these statistics are like for every 100 people that use this method, that's how many times it will be effective, 24 out of 100. As we go up this chart, again, some of these are, I'm sorry, I was incorrect. It's 24% of the time it's ineffective. It doesn't work. Condoms, about 18%. Going up higher, the hormonal methods, birth control pills, injectables, a hormone patch or diaphragm, those are in single digits, most of those. And then things like an IUD, vasectomy, or tubal ligation, those are under 1% failure rate. So those are very, very effective. Questions about the contraception section? Let's finish up with this. So sometimes the genetic counselor or an obstetrician can do what's called make a karyotype. A karyotype is basically they take a picture of the chromosomes a person has and kind of then cut them out and align them by size, longest chromosomes to shortest chromosomes. It can be made while the fetus is still in utero by sampling amniotic fluid. It shows a picture of the chromosomes that a person has. So the typical number of chromosomes in a human is what? 46 in a typical human body cell. And so here are some examples of karyotypes. These are pictures of unreplicated chromosomes. And we can see they're labeled by pairs, numbering 1, the longest chromosome, all the way to 22, which is the shortest chromosome. And so you can see them. They stain them, and they take up this certain banding pattern that's distinctive, and the banding patterns would match. So here we see an individual with their chromosomes. Also, you can look at the sex chromosomes. So there's one set of chromosomes. They're typically the 23rd. They're called sex chromosomes. And there can be X sex chromosomes or Y chromosomes. And these are one of the factors that play a role in the development of biological sex. And so the X chromosome is a large chromosome. We can see down here. The Y chromosome is actually a much smaller chromosome. Carries very little genetic information on it. This is what the Y chromosome looks like. It's just a tiny little thing. And so a person with XX chromosomes most commonly is a female. A person with XY is most commonly male biological sex. There's lots of variations here as well, that people can be born with 2 X's and a Y chromosome or only an X. There's a bunch of different what we call intersex conditions related to chromosomes, which you can see those things in a karyotype like this. You can also see if there's any other chromosome abnormalities like trisomy 21, which is more commonly known as Down syndrome. When a person has an extra 21st chromosome, then that leads to what we call Down syndrome, which is a collection of sort of characteristics that are typical when a person has an extra chromosome. You might think like, well, one extra chromosome, what really does it matter? Well, that extra chromosome makes extra proteins, which impacts sort of lots of different characteristics a person may have. There's other, Down syndrome is by far the most common trisomy condition when you have 3 of a certain chromosome, but there are others. There's trisomy 13. There's not a ton of them because often the effects are so great of having extra chromosomes that the embryo and fetus doesn't develop successfully and so doesn't survive. And so there's not situations when a person's born with 4 or 5 extra chromosomes because it results in an embryo that's just not viable. It's not going to normally grow and divide and form a fetus. But you could see those things in a karyotype like this. Here, this is the karyotype of a person that has Down syndrome. Right? And we could see, if we look at all these chromosomes, there's 2 of each one, but then we look at chromosome 21, and we see that there's 3 of them. How would a person end up with an extra 21st chromosome? Anyone have an idea about that? Or even, it's related to what process that we've learned about? You're close, John. Yes. It's something that happens during meiosis. If you think back to meiosis, the homologous pairs of chromosomes line up with their partners in the middle in metaphase 1. And then what happens to the partners? Typically, in anaphase. Well, they can cross over, and then what? What do the spindle fibers do? They split them apart, right? So can you guess how a cell would end up with an extra chromosome? If that doesn't happen, if the 2 homologous chromosomes, rather than splitting apart, they both go to one cell, the end result is that cell that comes about is going to have an extra chromosome in it because those 2 chromosomes didn't each go to separate cells. That's called non-disjunction. And so typically this increases in likelihood as a person gets older. So the older that parents are when they have a child, the greater risk of Down syndrome or some other non-disjunction difference. So it's because those chromosomes don't split up in the normal way. Again, here's some other examples. This is trisomy 21, Down syndrome. We can see it there. Here's another condition. This is 2 X chromosomes and a Y. This is called Klinefelter syndrome. A person has 2 X and a Y. There's other things that can happen. Sections of chromosomes can split, and crossing over can happen between 2 different chromosomes, which prevents some other issues. But any questions about any of that? All right, let's stop there. You want